Welcome 12th class students, Mrs. Nanda biology tutorial. Today I will start the next chapter of the genetics. I've already put 3 chapters complete for 12th class, that is reproduction in organism, then sexual reproduction in plants, and the first chapter principle of inheritance of genetics. You can observe I've already put it. So welcome the new 12th class session and see the next second chapter, molecular basis of inheritance of the genetics. Again, it is a tough, but you can learn very easily. I will explain in a very simple way. So molecular basis of inheritance, that means one material called genetic material, that is DNA. Full form is called deoxyribonucleic acid. Mostly the organism having DNA, which is a double standard. We will explain now the structure. But some organism also having RNA as a genetic material. But in mostly the virus, but RNA is not as stable. It remains unstable. So DNA as a stable genetic material. So next we will discuss the experiment also. Before discussing, we will discuss the various structures present in case of the DNA. So DNA is the main genetic material, play a role. What is the function of DNA genetic material? Help in information, carrying information. Again, they replicate. That is another feature and function. Replicate means give their own carbon copy. Third, again play important role in metabolism. That means DNA play important role. DNA give rise to RNA, which is the main part and take part in protein synthesis. It also play important role, that is in differentiation. Then the gene therapy, now these days, again this play important role DNA. So DNA present in both prokaryotes and eukaryotes. In case it is present prokaryotes, it is circular form. In eukaryotes, it is linear. In certain bacteria, these DNA, extra DNA present, which is also called plasmids, which take part in conjugation, extra DNA. But the DNA is the main part and it consists of polymers of nucleotides. So it's a long structure having the various base pair we will discuss now, and the DNA of the human genome, that is in haploid form 3.3 into 10 raised to power 9 base pair, that is a structure. Now, the structure of the DNA, which acts as a genetic material carrying various information from generation to generation. So this DNA further consists of the various structure. One is important structure is nucleotides, which further consists of two main structure, purines and pyrimidines. Purine and pyrimidine. Second, they are also having the sugar. And in this case, sugar is deoxyribose sugar. Deoxyribose. Then they are having the very important phosphate group. Phosphoric acid. Phosphate group, which is the main structure. Now, in this case, the first model or discovered, the various scientists discovered, but Watson and Crick got Nobel Prize and he gave the DNA model. And which model? So DNA according to him is first point double helix structure and consists of two strands. Clear? So double helix structure. The most common structure of the DNA in the organism is B-stranded. B DNA. That means spiral is right side going. So these are double helix structure, that is 5:3 and 3:5. This is the polarity. Both strands show the different polarity and they are complementary to each other. Complementary. Next, they run anti-parallel. They are anti-parallel to each other. Now these DNA double helix structure discovered by Watson and Crick. So in this case, it consists of one structure is nucleotide, then base pairs. So these are the base pairs and they are specific. Now these nucleotides consist of purine. First is purines, and purines are adenine and cytosine. Purine are these. Pyrimidines are adenine and guanine. These are the structure G. Cytosine, thymine are pyrimidines. So they show the pairing and they are present in case these structure. For example, adenine always pair with thymine like this. These are the base pair. Then guanine or cytosine always pair with guanine pair with cytosine or cytosine with guanine. So it means this is the group discovered by the scientist and they are having one group bDNA having 10 BP. BP means base pair. They are specific. For example, what is the structure? Adenine always pair with thymine by means of the hydrogen bond. Cytosine is always pair with guanine by means of the triple hydrogen bond. Next, these structures are further joined by means of sugar. Sugar deoxy, same sugar. In between, they are joined by means of the phosphate structure. So that means the structure same here. They are joined together and they constitute the chain. Now, you can see, you can observe adenine, thymine, cytosine. So you can learn the structure also side by side if you want to draw. But if you write the symbol, you get the credit. So it means sugar and phosphate. They are joined together and they form a complete chain, complete structure like this. Joining this structure. These diagrams are given in NCERT also. Now in this case, so adenine, thymine joined with this is the hydrogen bond. This joining is by means of glycosidic bond. And further they join by means of phosphodiester bond. So it means this is the complete chain. Same similarly like this. So this is the one part. So how many? So 10 base pairs are present in one group of this major and minor group, and polarity is different. Antipolar complement means adenine always pair with thymine by double bond. Cytosine always pair with guanine with the triple hydrogen bond. So they are specific. Same as sugar group, phosphate group. So they are equal. This is given by the scientist called Chargaff rule. The scientist who gave this statement, they are specific. They are and that rule all are same. Chargaff rule. So question can come for 2 marks. What is Chargaff? So it means thymine, adenine, they are same. Now, adenine with thymine. Similarly, cytosine with guanine. But in case of RNA, thymine is replaced by uracil. Thymine is replaced by uracil, that is adenine pair with uracil. That we will discuss in the next video, that is a uracil in RNA. So come to the point. So that means this is a structure and they are form the complete and they play very, very important role for the body, DNA. Then comes the prokaryotes and eukaryotes, and the topic according to NCERT is called packaging of DNA helix. Now in prokaryotes, it is very simple. Genetic material that is very simple. It is from network structure and they are not associated with histone proteins. So they are called nucleoid. It is also called incipient nucleus. It is also called genophore. So they are very simple. Prokaryotes means bacteria or the lower organism. They are very simple structure and their DNA that is uncircular, not associated with histone proteins. They form a very simple orb that is called nucleoid. Similarly, in eukaryotes, in the higher organism, this packaging of helix, that means consists of DNA is associated with histone proteins. Histone proteins. So they are 5 in number: H1, H2A, H2B. Now, this is and they constitute octamer. What is octamer? This is the H1 structure. This is octamer, 4 inside. That means H2A pairing, 2H2A, and similarly 2H2B. So 2 constitute for octamer. Otherwise, they are total 5, and 1 H1 remain outside and wrapped by DNA, which is a negatively charged. Wrapped by this DNA structure, so they form a complex structure with histone proteins, and they are 5 in number, and they are formed octamer like this inside. They are 8 in number inside, and 1 is outside, constitute the packaging material of DNA helix. Similarly, this DNA is also in the form of chromatin material, network structure, genetically. 2 types, euchromatin and heterochromatin. And they appear as in beaded structure like this and called nucleosome. This is the diagram of nucleosome given in your NCERT. That is nucleosome structures. I have drawn this, and this is nucleosome, the beaded structures. Nucleosome, beaded structure. So they consist of 2, euchromatin and further heterochromatin, and this euchromatin are in the loosely packed, light stained, and heterochromatin are dark structures, right? So euchromatin are more active than heterochromatin, so that means they play very, very important role. In the next video, we will discuss that DNA is the main genetic material, not RNA, explained by the various experiment. Up to here, I think the topic is clear to you. Thank you.